Hi, I'm Kayla. It is May 2nd, 2024, and today I will be presenting two case studies on different individuals. The first case is based on the mechanisms of ammonia throughout the body, and the second case will be on the explanations of drugs that reduce plasma cholesterol.
For our first case study, we have a newborn infant who has developed lethargy, hypothermia, and apnea all within the first 24 hours after birth. Her blood work showed that her initial blood ammonia levels were elevated at 314 and rose rapidly to 600. Based off these results, she will more than likely be diagnosed with hyperammonia. For reference, a normal blood ammonia level ranges between 17 and 80. We need to find out why she has had such high levels of ammonia in her blood. To do that, let's investigate the liver for possible dysfunction. Let's do an overview of a hepatocyte cell within the liver. The liver plays a crucial role in ammonia metabolism. Ammonia is a toxic byproduct of amino acid metabolism and various cellular processes in the body. Within the mitochondria of the hepatocyte, ammonia NH4+ undergoes a series of steps and mechanisms that take place to convert into urea. Before ammonia is excreted, we begin with amino acid catabolism. In box A, we are shown that we get most of our amino acids through our dietary protein intake. The excess of amino acids are utilized for protein synthesis, nitrogen compounds, and amino acid oxidation. In box B, the amino acids undergo processes such as transamination and dehydrogenation. Transamination is an important process in amino acid metabolism. It reduces the concentration of ammonia in the body by transferring and forming a new amino acid group. Amino acid groups are transferred to a keto group, which results in the creation of a new keto acid, for example, glutamate. It also serves as an excess ammonia disposer. However, some of the free ammonia is expelled through our urine, also known as urea, which is a byproduct of protein digestion. Now let's discuss glutamate dehydrogenase. Glutamate dehydrogenase is an enzyme that catalyzes the reversal conversion of glutamate to alpha-ketoglutarate using either NADH or NADPH, therefore producing ammonia in the process. When ammonia levels rise, glutamate dehydrogenase facilitates the removal of excess ammonia by converting it to glutamate. Moving on, glutamine synthase is an enzyme that is primarily found in the liver. It converts ammonia and glutamate into glutamine. Glutamine is then transported from the liver to the kidneys. It is then metabolized by the enzyme glutaminase, releasing ammonia for excretion in the urine. Lastly, we'll discuss the role of glutaminase for this portion of the case study. Glutaminase is predominantly active in the liver and catalyzes the hydrolysis reaction of glutamine to produce glutamate and ammonia. The free ammonia that is generated is incorporated into the urea cycle to be detoxified. When in a fasted state, glucose levels decrease and glycogen stores are depleted. The body must undergo glycogenesis to produce glucose for energy. During this time, amino acids are released throughout the body to be utilized as substrates for glucogenesis. An important amino acid would be alanine. Alanine is produced when amino acids for glutamate are transferred to pyruvate. Alanine undergoes transformation to regenerate pyruvate, which is transported to the glucogenic pathway to produce glucose. This is known as the alanine glucose cycle. This cycle facilitates the transport of nitrogen. After reviewing the mechanisms of ammonia detoxification, the infant's increased levels of uric acid indicates a defect in the urea cycle. I think that the enzyme that is most likely to be deficient would be the carbonyl phosphate synthetase, or CPS. CPS is a crucial step in the urea cycle because it is the first step in detoxifying ammonia within the body. If this enzyme is deficient, then an individual would suffer a number of issues such as what the infant is showing, aka hyperammonemia. If left untreated, CPS deficiency can lead to severe health complications such as brain damage or death. The least likely enzyme that will be impaired is arginine. Arginine is utilized in the last step of the urea cycle. Therefore, it would not cause uric levels to elevate. Arginine is technically a non-essential amino acid. However, it can become essential in times of dysfunction in human growth and development. Arginine is a precursor for the synthesis of ornithine, which helps maintain nitrogen balance and prevent the buildup of excess ammonia in the body. On to the second case study. We are presented with a middle-aged female that is having chest pains and worsening fatigue on exertion for the past 3 months. After taking her vital signs, her blood pressure was elevated at 160/80, a pulse rate of 80, and respiration at 12, while only being 5'5.5", and weighing 250 pounds. Based off of this information, she would be considered obese for her height. Her fasting blood glucose levels were 221 and 196 while fasting. Normal levels are between 70 and 150. Her lipid and A1C panels were elevated as well. The female patient is most likely experiencing a resistance to insulin. When we are in a fasted state and our blood glucose levels are elevated above the normal range, our bodies are producing insulin, but in her case, her body is not responding to it. With her glucose uptake being reduced due to impairment, she will be diagnosed as hyperglycemic. This also explains why her lipid panels were elevated as well. When there is insulin resistance, free fatty acids are released from the surrounding adipose tissues and into the blood, therefore leading to elevated triglycerides, high LDL cholesterol, and low HDL cholesterol. The possible treatment plans that were presented by her physician suggest that she tries statins, PCSK9 inhibitors, and bile acid sequestrants to lower her plasma cholesterol levels. Statins are a class of drugs that inhibit the enzyme HMG-CoA reductase. This in turn will reduce the production of cholesterol in the liver.
PCSK9 inhibitors, also known as proprotein convertase subtilisin kextin type 9 inhibitors, are proteins that regulate LDL receptor levels on the surface of liver cells. Once binded to the receptors, the liver will not be able to remove LDL cholesterol from the blood as efficiently. The PCSK9 inhibitors blocks those receptors, so the liver is able to remove LDL cholesterol. The last treatment option is taking bile acid sequestrants, which is also known as bile acid binding resins. Bile acids serve to absorb dietary fats and cholesterol in the intestine. This medication blocks the enterohepatic circulation of bile acids, so there will be less buildup in the body, and most of the bile fats will be excreted through bowel movements. This will definitely help to lower LDL cholesterol, which is also known as the bad cholesterol.